Part 2 of Lab 3 looks at cell function. More specifically, we're looking at how substances can move in and out of a cell. And there's two different processes that substances can use to move from one place to another. One is called diffusion, and the other thing that we're going to talk about is osmosis. Osmosis refers specifically to the movement of water, whereas diffusion refers to the movement of other stuff. So you can see that diffusion is really important in a lot of different natural processes. For example, how does our body take up oxygen from our lungs? It's actually through diffusion. Oxygen diffuses into our capillaries, and carbon dioxide diffuses out of our capillaries into our lungs where that gas is then expelled. Diffusion is also important in the absorption of nutrients. For example, here you can see the inside of the intestines, and these little yellow particles are different types of nutrients. And some types of nutrients can actually just simply diffuse, and they'll just naturally move out of the intestines and into the capillaries. Before we get into talking about diffusion and osmosis, let's talk about the difference between a solute and a solvent, because you'll see these words a lot. A solute is the dissolved particles. So for example, if I were to mix together salt and water, the particles that are actually dissolved in there is the salt. Okay, so the salt is the solute. I always think of it as it's the stuff in there. Okay, the solute is the stuff. The solvent is the liquid that's doing the dissolving. And in this case, the solvent is water. So the water is dissolving the salt. Okay, the universal solvent in biological systems is water. So water is a very important solvent. It dissolves a lot of the nutrients in our body and so forth. When we talk about the solute and the solvent together, whenever they've mixed, we create a solution. Another concept that you need to understand is this law of Brownian motion. It states that the random movement of particles suspended in a fluid, such as a liquid or a gas, are the result of collisions with other particles. Brownian motion was first observed when a scientist was looking at pollen grains, as you can see here on the right in a slide. And it almost looked like the pollen grains were swimming around or moving. So he thought they were alive and just swimming in there. What we realize is that they're actually vibrating because all of the molecules within that solution are actually just bouncing off of each other because they're constantly in motion. So particles will constantly be colliding with each other, whether there's another force acting on them or not. The state of matter of a substance is actually determined by this motion. So if something that's a solid is very dense, the molecules are really close together, so they don't have much room to move. Whereas in a liquid, the molecules can move a lot more. And in a gas, they can move even farther and faster before they even collide with each other. So now let's talk about diffusion. Diffusion is specifically the movement of molecules, and we're talking about the solute, the stuff in there. So diffusion is the movement of the solute from an area of high concentration to an area of lower concentration. The solute will always move down its concentration gradient to an area where there's less of it. And that's just the natural way things move. And the reason that happens is that there's a lot more collisions happening here when there's a higher concentration, causing these particles to bounce in this direction where there's less stuff. You can see this if you were just to pour, for example, red dye into a beaker of water. You would see the dye eventually start moving to the area where there's no dye until it's distributed evenly throughout that solution. Here you can see that there's a lot of particles over here on the left starting out, and the net flow is to the right, and eventually the flow to the right and the left equals out. And there is no net flow. Diffusion explains how particles can move from, let's say, one side of the cell. So this is the extracellular space, so it's the space outside the cell. This is the intracellular space down here. If there's a lot of a substance outside of the cell, it can simply diffuse down its concentration gradient into the cell. So what factors can affect the rate of diffusion, so impact how fast something moves from one place to another? You'll be testing a lot of these factors in lab. The first factor is temperature. The second factor is type of medium. So is the solute trying to move through a solid? Is it trying to move through a semi-solid, a liquid, a gas, and so forth? And also the particle size. So the size of the solute can affect how fast it can diffuse. You will not actually be testing temperature in lab, but you will be testing the other two factors. But you should still know the effects of temperature on the rate of diffusion. Temperature is actually the measure of kinetic energy of a system. So it's measuring how fast the particles are moving. So if you increase the temperature, it means the molecules are moving faster and colliding more. You've seen this when you've tried to make tea, for example. If you put a tea bag in hot water, the tea diffuses outside of the bag really quickly. However, if you try to brew tea in cold water, it takes much longer for the tea to diffuse out of the bag, and that's because of the colder temperatures. Here is a little experiment. On the left is showing hot water, on the right is showing cold water. And the experimenter is going to add some food color into these two beakers, and you'll be able to just see the movement within the hot water is much greater because the particles are colliding much more. Notice that diffusion is happening at a greater rate in the hot water than in the cold water. Let's look at the effect of the type of medium on the diffusion of a solute. So here you can see three different types of medium: a solid, a liquid, and a gas. And if a solute were to move through each one of these, which one do you think will move at the fastest rate? What you would expect to see is that the less dense the medium, so as you go from a solid, liquid, or to a gas, the faster the rate of diffusion. This should occur because it's easier for particles to move through a less dense medium. You will also look at the effects of particle size on diffusion rate. So imagine this is a cell membrane, and here's a pore in the cell membrane. When you have these little openings or channels in the cell membrane, and certain particles can fit through and not other particles. So smaller particles typically will have a faster rate of diffusion than larger particles, and extremely large particles may not even be able to move through a membrane if they can't fit through the pores or the channels in that membrane. You will be testing the effects of particle size and diffusion rate during lab. In order to simulate a cell membrane, you will be using a dialysis tube. Dialysis tubing is similar to the cell membrane in that it has tiny little pores throughout the membrane, and it's selectively permeable, which means some things can go through the membrane and other things cannot. You will be filling this dialysis tube with a solution that contains both glucose and starch. And surrounding the dialysis tube within the beaker will be a solution containing iodine and water. Remember that glucose and starch are both carbohydrates. Glucose, however, is just a single sugar molecule, whereas starch is made up of many glucose molecules linked together to form a long chain of sugar molecules. So starch is a lot bigger than glucose. You will test for the movement of starch by looking for a color change. Remember that when starch and iodine interact together, it turns black. So if iodine enters the bag where the starch is, the inside of the bag would turn black. If starch leaves the bag and goes into the iodine, then the iodine solution on the outside here would turn black. The way that you're going to test for glucose is actually using a glucose test strip. This test strip, so remember glucose is starting in the bag, and you're going to test to see if glucose has left the bag. So you'll test this brown looking solution out here. You'll stick this test strip in there, and if the test strip changes color, that means glucose is present and it has left the dialysis tubing. Now let's move on and talk about osmosis. Osmosis is the movement of water, and only water, across a semi-permeable membrane to an area that has a higher solute concentration. Water will always move from an area that has a lower solute concentration, so that means there's less stuff in it, to an area that has a higher solute concentration, so there's more stuff there. We give a fancy word to a solution that has less solute in it, and it's called hypotonic. Hypo means less. A solution that has more solute in it is called a hypertonic solution. Hyper means more. So a lower solute concentration is hypotonic, a higher solute concentration is hypertonic. Water always goes from hypotonic to hypertonic. A lot of times I tell students, don't just memorize something, you should know why it happens. In this case, I want you to know why it happens, but I also want you just to memorize this. It will help you a lot. So remember, water always goes hypo to hyper. If you remember that and memorize that, it will make solving a lot of these problems easier. So let's look at an example here. Let's say that I have this selectively permeable membrane in the middle here. The left side here has less solute, represented by these purple circles. The right side has more solute. This has less solute, so it's called hypotonic. This one has more, it's hypertonic. Water always will go to the area that has more stuff in it. So if we just let this sit here for a while, we would see that the water level on the side with more solute will actually increase, and the water level on the side with less solute will decrease as the water moves to the right. Why does this happen? Well, let me explain how this happens by looking at this diagram here. The white molecules are the water. The dark circles are representing the solute in this case. So on the left side, we only have one solute molecule, and water tends to interact with the solute. So here you can see the water molecules surrounding the solute and interacting with it. When there's less solute in a solution, there's more free water molecules. So there's more water molecules that are capable of moving. So these water molecules are more likely to move to the other side. Whereas in this hypertonic solution, where there's a lot of solute, notice there's not very many free water molecules. So there's less water available to move to the other side. So that is why water will typically leave a hypotonic solution. Definitely know these terms that describe tonicity. If I ever ask you to describe the tonicity of a solution, I'm asking for you to use the words hypotonic, hypertonic, or isotonic. Those are the different types of tonicity. Hypertonic again means that the solution has a higher concentration of solutes than the other solution it's being compared to. Hypotonic means it has a lower concentration of solutes than the other solution it's being compared to. And isotonic means that the two solutions have an equal concentration of the solute. So there's going to be no net movement of water in either direction. Water will still move in both directions, but at an equal rate. Let's do some practice predicting the movement of water in various solutions. So I have 3 bags inside this beaker. Each bag contains a different concentration of salt. The outside of the bag, so the solution in the beaker, contains 30% salt. The bags are not permeable to salt in this case, so salt can't go in and out of the bags, but we're just talking about the movement of water here. In the first bag, in this first case here, it contains 50% salt. The outside contains 30% salt. Will water go into or out of the bag? Remember that water always moves to an area with a higher solute concentration, so water will go into the bag. So is the inside of the bag hypertonic or hypotonic relative to the outside of the bag? Remember what I said to memorize. Water always goes hypo to hyper. So since the arrow is pointing this way, the outside of the bag is hypo, the inside of the bag is hypertonic. Also, hyper means more. So this is the area that has a higher solute concentration. In the second case, the inside of the bag only contains 5% salt. So what will happen here? Well, water will move out of the bag into the area that has the higher solute concentration. So in this case, the inside of the bag is hypotonic relative to the outside of the bag. In the last case, there's an equal concentration of salt in the inside of the bag and the outside of the bag. So water will move equally in and out of the bag. And we would say that the inside of the bag is isotonic relative to the outside of the bag. So the best way to practice with this is to just keep practicing doing practice problems like this over and over again. You could come up with your own examples and test yourself and make sure that you're just understanding this concept. Another thing to make sure that you don't make mistakes when you're trying to look at osmosis and diffusion is if I'm talking about the movement of water, then I'm talking about osmosis, okay? However, if I'm talking about the movement of a solute, I'm talking about diffusion. This can get confusing because water always goes to an area that has more solute. But if I was talking about the solute itself, a solute always moves down its concentration gradient. So let's look at the same example here, but instead I'm asking about the movement of the solute. So we know water would go into the bag in this case, but if salt could move, where would salt go? Salt would move out of the bag, down its concentration gradient, from 50% to a 30% solution. So if you're not paying attention and you're reading a problem really quickly and you're not paying attention to whether I'm asking about the solute or the solvent, which is the water, then you're going to get this problem wrong because they move in opposite directions. The water's moving in, trying to balance out the two solutions, whereas the salt's moving out, trying to balance out the two solutions. Right? So they're trying to create a balance here. So if the salt moves to the area where there's less salt, it can almost balance the two sides. But water could do the same thing by moving into the area where there's more salt to try to balance the two solutions. Okay? Hopefully that makes sense. In the second case here, where would salt move? Well, there's more salt outside the bag, so the salt itself would move into the bag in the opposite way of the water. In the last case here, the salt would move equally in both directions because we already are at an equilibrium. So why is this important? Why are we talking about osmosis and the movement of water in and out of cells? Well, this is really important because animal and plant cells require specific environments that give them their ideal conditions. For example, animal cells want to be surrounded by a solution that's isotonic to the environment within the cell. So water is going equally in and out of the cell. So here's a picture of a red blood cell. This is the normal condition of a red blood cell, and this is how it wants to be, pretty much. Whereas a plant cell, if the outside of a plant cell has an equal solute concentration as the inside, the plant cell will actually become kind of flaccid. The vacuole won't be filled with water, so it's just going to be a little bit limp. However, what will happen if that plant cell and animal cell were placed in a hypertonic solution? So the outside of the cell was hypertonic relative to the inside of the cell. Well, if you remember what I said before, water always goes from hypo to hyper. So if a cell's placed in a hypertonic solution, that means water will leave that cell. So what happens to a plant cell if it's placed in a hypertonic solution? Well, all the water would leave the central vacuole. And so here you can see the central vacuole is really small. The cell membrane shown in this yellow color here, and you can see the cell membrane actually pulls away from the cell wall. And the cell wall in this case would start to kind of cave in a little bit too. We call this plasmolysis whenever a plant cell is placed in a hypertonic solution. When a plant cell experiences plasmolysis, it begins to wilt because the vacuole loses all of its water. An animal cell would experience something similar in that the water would leave the cell and it would become all shriveled up. The term we use to describe an animal cell in a hypertonic solution is crenation. If we were to place a plant cell in a hypotonic solution, so the outside's hypotonic, that means the inside's hypertonic. If one solution's hypo, the other one has to be hyper. So they have to be the opposites of each other. So if the outside's hypotonic, we know, based on what I said to memorize, water leaves a hypotonic solution, so it would go into the cell. This is actually the condition plants like the best, because this means that water enters the plant cell, the vacuole gets swollen, and it pushes out on the cell wall. This creates what we call turgor pressure within a plant cell. And because the cell wall of a plant cell is very rigid, it prevents the cell from bursting. And instead, it makes a nice crispy vegetable. All right, so a plant cell that has a lot of water in it will be very full and very tall and crispy. However, animal cells cannot survive in a hypotonic solution because they do not have a cell wall to prevent them from bursting. So if an animal cell is placed in a hypotonic solution, so that means that the outside of the cell has less solute, water will go into the cell and it will cause it to burst. And when a red blood cell or an animal cell bursts, we call this hemolysis. Lysis means to break or to burst, so the cell is being lysed in this case. Now I'm going to show you a video of the effects of a hypertonic and hypotonic solution on blood cells. What's shown in gray up here are red blood cells. What's shown in white here are white blood cells. And you'll be able to see that the effects of them first being placed in a hypertonic solution, they'll start to shrivel up. Then in the end, they'll be placed in a hypotonic solution. So water will enter these cells, and in the case of the red blood cells, they'll actually disappear because they'll actually slowly start leaking because they just got filled with too much water, and they'll just break apart. Whereas you'll see with the white blood cells, they'll actually burst and you'll see all their contents come out because they just can't take all the water that that's being taken in. So here they're being placed in a hypertonic solution, so they're starting to shrivel up. The effects aren't really obvious in that case. Now they're placed in an isotonic solution, and now they're being placed in distilled water. So distilled water is hypotonic. There's no solutes in there. So you saw the red blood cells disappear, and now the white blood cells are filling with water. They're expanding, and eventually they won't be able to contain that anymore, and they'll burst. So it's very dangerous for your cells to be placed in an environment like that. So if your body can't regulate its solute concentrations very well, it can be very disastrous. The final thing that we're going to be looking at in this lab is the effects of acids and bases. When we measure the acidity of something, we usually use a scale called pH. pH is a scale from 0 to 14 that specifies the hydrogen ion concentration in a solution. So the more acidic a solution is, the more hydrogen ions it has. The more basic it is, the less hydrogen ions it has, and the more hydroxide ions it has. So it's the balance of these hydrogen ions and hydroxide ions that determines how acidic a solution is. A solution that's neutral, has a pH of 7, would have an equal concentration of hydrogen ions and hydroxide ions. So again, acids contain a lot of hydrogen ions, so something that is considered an acid would release a lot of hydrogen ions into the solution, and therefore would have a pH less than 7. So the smaller the number, the more acidic it is. Something that's considered a base would actually remove hydrogen ions from the solution, or in other words, it would release hydroxide ions. And it would also have a pH that's greater than 7. The higher the pH, the more basic it is. Here are some examples of some solutions and how acidic and basic they are. You can pause the video and study these if you would like. Some of the most acidic compounds are hydrochloric acid, and some of the most basic ones are sodium hydroxide. Some things that are rather neutral would include pure water and tears. Our body has to maintain a specific pH for it to function properly. And we naturally produce compounds called buffers that actually resist changes in pH by adding or removing hydrogen ions. If, for example, our blood contained too many hydrogen ions, that buffer could remove the hydrogen ions. Or if it didn't contain enough hydrogen ions, it could release hydrogen ions into our blood to help maintain a specific pH. Therefore, organisms have natural buffers to prevent pH changes. Our normal pH level is between 7.35 and 7.45. If our pH even drops below 7.35 or above 7.45, we would start to feel the effects of that. And if our pH in our blood drops to 7 or below, we could die. And above 7.8, we could die. So our body naturally produces a buffer called carbonic acid that actually can release hydrogen ions or take up hydrogen ions in order to regulate pH. Notice that buffers do not always maintain a neutral pH, right? For example, in our blood, we're not trying to keep a pH of 7. We need our pH to be slightly above 7. Buffers are usually very specific, so there might be some buffers that might keep a solution more acidic, some buffers might keep it more basic. The main point here is that buffers keep the pH from changing from a certain level. Thanks for watching, and that is the end of the background information for the third lab.